Okay, so earlier we talked about the glucose pyruvate pathway and how this plays into or is connected to the urea cycle. And so I want to pick that back up now and talk a little bit more about that. So, glutamate that we just made in the process of removing ammonia from those amino acids. Glutamate can actually donate that ammonia to pyruvate to make alanine. So that's what we want to talk about a little bit now. This is called the glucose alanine cycle. And this is an important pathway in your cells in order to, when we're thinking about nitrogen excretion. So we know that vigorously working muscles can operate near anaerobically conditions and rely solely on glucose for energy. So that is if we're working out, if we're, during periods of heavy exercise when our oxygen supplies might be depleted, those muscle cells can operate in an anaerobic condition and produce large amounts of pyruvate. We know that if the pyruvate is not eliminated from the muscle cells, that's when we get that lactic acid buildup through lactate fermentation. That's when you get that sort of burning sensation in your muscles. Now, alternatively to that, what can happen is the pyruvate can be converted to alanine for transport to the liver. That would prevent that pyruvate from building up inside your cells and be converted to lactic acid. This is for people that can exercise for long amounts of time and not get that burning sensation in their cells. And this is why they can do that, is because that excess pyruvate is not being converted to lactic acid and building up, but it's actually being converted to alanine and transported to the liver. We also know that alanine functions in this role as a way to transport those amino groups to the liver in order in another non-toxic transport form so that we can process those amino groups for excretion. Okay, so this is the glucose alanine cycle. So we're going to go over exactly what happens here. So we're going to start up here in our muscle protein. So we're if we are again during activity, vigorous exercise, we might degrade that muscle protein and start breaking down those amino acids for fuel. So we've broken down our proteins into amino acids, and then we have those amino groups that we separate out, and we're going to transfer those to glutamate. Because remember, that's the goal. When we break down those amino acids, we're going to separate off the carbon skeleton from the amino groups, and we transfer those amino groups to glutamate. That is the goal for nitrogen processing. So what happens is that glutamate, we know a couple of things can happen to the glutamate. It can be converted to glutamine and transported directly to the liver. So we could take this glutamate, transfer it, or convert it to glutamine and send it directly to the liver for processing. Or we can enter into this glucose alanine cycle where we have a conversion of the transferring of that glutamate to pyruvate and then to alanine. So what happens here is we can have a alanine aminotransferase. So we're transferring that amino group now to alanine. So we take it off of the glutamate. Pyruvate is the acceptor here. So pyruvate accepts the amino group and is converted to alanine. So we're left here with the alpha-ketoglutarate. Well, alanine can actually enter the bloodstream and travel to the liver. So we have alanine is now in the blood, and now alanine can be distributed or deposited in the liver, where now the amino group is going to be transferred from alanine to alpha-ketoglutarate. So alpha-ketoglutarate accepts the amino group, so we have another aminotransferase here that's going to function to transfer that that amide group back to alpha-ketoglutarate to reform glutamate and pyruvate. So when we take that amino group off of alanine, we convert that back to pyruvate. Pyruvate can be converted back to glucose, we know, through gluconeogenesis. So remember, that's sort of a energetically expensive process that if we have to carry that out, we want that expense imposed on the liver and not the muscles. We don't want the muscles having to expend their energy to convert pyruvate back to glucose, but the liver can do that. So remember, gluconeogenesis is going to happen predominantly in the liver. So this is converting that pyruvate back to glucose. Glucose can then be transported back through the bloodstream back to the muscles, and then we can use the process of glycolysis, right? We know glycolysis converts glucose to pyruvate as a way to produce ATP for those muscle cells so that it's available for contraction. Now, I want to go back here to this point real quick in the cycle. So we transferred that amide group from alanine to alpha ketoglutarate, and we got glutamate. So then glutamate can then be used in the urea cycle to take that ammonia and take that amide group and transport it to urea. And so that's how we can excrete the excess nitrogen. So we'll circle back to this cycle in just a minute and talk a little bit more about that urea cycle. But this is one way that your cells process the breakdown of amino acids when we are using muscles for fuel. Okay, so we've touched on this before. The problem with nitrogen excretion and nitrogen processing is ammonia. So free ammonia is highly, highly toxic. And so we have to be cautious and careful on how we utilize these amide groups, how we extract them, how we excrete them, that we don't end up with free ammonia. So when ammonia is released from glutamate in humans and other vertebrates, it is directly converted to urea for excretion. If free ammonia were to be allowed to be free in the bloodstream, that could cause a lot of problems. So again, ammonia readily crosses the blood-brain barrier, and it can induce a comatose state. It can cause swelling in the brain, so cerebral edema. It can increase that cranial pressure. It can result in potential loss of ATP in the brain cells. So ultimately, death. If you have significant ammonia toxicity in your bloodstream, it's ultimately going to result in death. It is especially harmful in developing brains. So in the fetus, it can cause extreme loss of neuron. It can alter synapse formation so that you're not able to make connections between your brain cells like you should. It can cause severe deficits in cellular metabolism, meeting the energetic needs of those cells, which again would pretty much ultimately result in death of the fetus. So ammonia toxicity is a very serious problem and something that we have to have a good way to avoid. And the way that we avoid that is really by the production of urea through the urea cycle. So the first step in this cycle is the carbonyl phosphate synthase 1, and that is what captures that free ammonia immediately in the mitochondrial matrix and causes it to enter the urea cycle. So this is the first step of the urea cycle. It's a highly regulated step, which we'll talk about kind of towards the end once we go through the cycle. But this is your body's way of avoiding that free ammonia in the bloodstream. Okay, so if nitrogen sources or these amide groups are not reused, and by reused I mean if they're not applied to biosynthetic pathways like the purines or the pyrimidine pathways and used as that nitrogen source, they're channeled into the excretory cycle, which in our case is the urea cycle. This is the way that we get rid of that excess nitrogen. Urea is ultimately going to be passed in the bloodstream and excreted through the urine by the kidneys. So this slide here demonstrates the first step sort of in the urea cycle, the production of carbonyl phosphate. So this is the first nitrogen acquiring step, the way that we sort of harvest or harness that nitrogen. So this is occurring in the liver, in the mitochondrial matrix of the liver. So the first thing that happens is bicarbonate. So we know we have bicarbonate in the bloodstream. This is going to be phosphorylated by ATP to produce this carbonic phosphate acid anhydride. So this is an intermediate along this step. Ammonia is actually going to be able to displace this phosphoryl group. So any free ammonia, whatever its source, whether it comes from the intestines, whether it comes from other tissues, whether it comes from amino acid breakdown, wherever this ammonia is coming from, and we've talked about several sources of this ammonia, wherever it comes from, it's going to be able to displace that phosphoryl group on the carbanic phosphoric acid anhydride to produce carbamate. So carbamate is then going to be phosphorylated again by ATP to produce this carbamyl phosphate. This is what actually enters the urea cycle. So once we have produced carbamyl phosphate, we are able to enter the urea cycle and actually excrete or get ready to prepare all of these amide groups, all of this nitrogen for excretion. Okay, so this is a little bit overwhelming slide. This is showing us where that nitrogen from the carbamyl phosphate is going to enter the urea cycle. So everything from here to here is what we've already been talking about in chapter 18. So this area down here that I'm highlighting in red, this is the urea cycle. So this is what we've been working our way towards all this time. So everything in the top of this slide, we have our amino acids up here, and so this is looking at that conversion of extracting the carbon skeleton from that and removing the amide group, putting that on glutamate, and then this is the mitochondria. So this little gray box here is representing the mitochondria. So this is showing us that conversion, that transport of glutamate into the mitochondria. And then the slide that we just talked about is showing that production of carbamyl phosphate here. So once we have produced the carbamyl phosphate, we are ready to enter the citric acid cycle. And I'm going to show you this in a little bit more detail in the next slide. I'm going to break this down and talk about the different steps of the cycle. But all of this, so everything up here has been working towards getting that production of glutamate. So remember, that is the point of all of this nitrogen processing that we have been talking about up until this point. It's taking all of that nitrogen, whatever the source, whatever the amino acid that it comes from, and getting it in the form of glutamate so that we can send it to the mitochondria to be able to produce carbamyl phosphate so that we can enter the urea cycle. Okay, so here we're ready to talk about the reactions of the urea cycle. So the first, and again, it's kind of a cycle, but you can so you could start wherever you want to. But I'm going to start up here at the top with our production of carbamyl phosphate that we just made. So the first reaction is the donation of that carbamyl group to ornithine. So here's ornithine. This is involved in the starting reaction of the urea cycle. So it's similar to oxaloacetate in the citric acid cycle, where that concentration is kept low and it's going to be regenerated through the use of the cycle. So what happens here is carbamyl phosphate interacts with ornithine. It's going to donate that carbamyl group to produce citrulline. So here we have the production of citrulline, and in doing so, we release a phosphate. So we're going to have a free phosphate group that is released here. Citrulline is an important intermediate here because it can pass from the mitochondria into the cytoplasm. So this first reaction of the urea cycle is taking place within the mitochondrial matrix. The rest of the urea cycle is going to take place in the cytosol. So citrulline is our bridge there. This is the molecule that can pass from the mitochondria into the cytoplasm. The second step of the urea cycle is kind of broken down into two steps. So we have that citrulline. We're going to take a second amino group that enters, that's going to come from aspartame. So, or aspartate, I'm sorry. So here we have aspartate here, that where we're going to be able to sort of harvest or capture a second amino group. So this is generated from a transamination reaction within the mitochondria. So here we have aspartate up here in the mitochondria. Aspartate is going to travel again into the cytoplasm, and we're going to be able to capture that amino group, which here is highlighted in green. and we're going to form arginosuccinate. So that's our intermediate from this step. This step requires ATP. So you can see here this step requires the breakdown of ATP in order to form this intermediate to get us to the arginosuccinate step here. The third reaction of the urea cycle is arginosuccinate is going to be cleaved to form arginine and fumarate. So we know fumarate, we know this is an intermediate that is used in the citric acid cycle. So this is going to be would be kind of cycled off or sucked off to be used within the citric acid cycle. And arginine is going to continue into or can continue along in the urea cycle. This is the only step 3, this is the only step of the urea cycle that is reversible. So this is a potentially reversible step. In the last step, arginine is going to be used to regenerate ornithine and also to produce urea. So we're going to cleave arginine in a hydrolysis reaction to produce urea and regenerate ornithine. So urea is our key here. Urea is what we are going to be extracting. We're going to send that to the kidneys, and it's going to be dissolved in the water and processed in the urine through the kidneys. Ornithine is obviously regenerated and reused to continue this cycle. So you are not responsible for knowing the enzymes or I wouldn't ask you to draw the steps of this cycle, but I would ask you to know and understand what's happening in each step, and we are going to talk a little bit about regulation. Okay, so the flux or the flow of the urea cycle can vary dramatically with diet. So it's very diet dependent, which is a little bit unique, or we see that a little bit with glycolysis as well. But there are a couple of regulation points along this cycle. So the first is that carbonyl phosphate synthase 1. So this is the enzyme that is responsible for producing the carbonyl phosphate, which is involved in the first step of the urea cycle. So this is activated by N-acetylglutamate. N-acetylglutamate is synthesized from acetyl-CoA and from glutamate. So we take glutamate, we take acetyl-CoA, we form N-acetylglutamate. In mammals, this molecule has a primarily regulatory function, or purely regulatory function. Actually, that's its only job, is to regulate the carbonyl phosphate synthase 1. When glutamate and acetyl-CoA concentrations are high, it activates the urea cycle. So that tells us that we have produced a lot of glutamate in the cell. Our acetyl-CoA levels are high, telling us that we probably had a lot of pyruvate being produced in the cell, and that tells us that we're going to have a lot of nitrogen that we are going to need to process. And so we need to activate this enzyme, the carbamate synthase 1 enzyme, and that the urea cycle is going to be going to increase its flow or increase its flux. So this high levels of glutamate and high levels of acetyl-CoA activate the urea cycle. We can also look at expression of those enzymes in the urea cycle. We didn't go over the names of each one of these enzymes, but there are 5 enzymes that are involved in this urea cycle. So when conditions require it, the expression of these enzymes is increased. So we're talking about when we're eating a high protein diet. So if you've ever gone on the Atkins diet or something like that where you're eating almost all entirely protein, the expression of these urea cycle enzymes would be activated, would be increased. Also during times of starvation, when we are starting to break down those proteins for fuel and for energy for our cells, and therefore we would have all of those amino acids that we would need to process, we would see increased expression levels of these urea cycle enzymes, knowing that with all of the amino acids that we're breaking down, we're going to have all of this nitrogen that we need to process, and we're going to have to increase the flow through the urea cycle. If you are eating a protein-free diet, so in cases of vegetarians or vegans who aren't eating a lot of protein, you would be expressing these enzymes at very low rates because you wouldn't have the amount of nitrogen processing and the amount of flow through the urea cycle that would be required at other times and other conditions. Okay, so then lastly, I thought this was an interesting chart from your book. This is just a chart that lists some human genetic disorders that are affected by or that affect amino acid catabolism. So You can look, for example, we have deficiencies in the carbamyl phosphate synthase 1. So this would obviously affect synthesis of urea. This could result in lethargy, convulsions, and ultimately even death. If we don't have the proper way to process that nitrogen and excrete that nitrogen, again, that can lead to ammonia toxicity, which is really a problem and could lead could lead to death. I think a couple of you, or at least one group, is doing the maple syrup urine disease for your metabolic disease process. So again, this is a defect in amino acid processing. So it's a way that we degrade isoleucine, leucine, and valine, and we can't degrade those amino acids properly. And so that can also lead to serious problems. So the phenylketonuria, this is also a pretty common metabolic disease that affects processing of phenylalanine in the body. So again, this is just an interesting little chart that talks about, or that demonstrates, all of the problems that can occur just from not being able to process urea or produce urea and process nitrogen normally in the cells. So that's the end of chapter 18. I will see you in class.